All right. Good morning, everyone. We have reached week 8. It is Monday, July 27th, 2020, and this is our last Monday lecture. Only 2 more lectures to go, and only 1 more section to go. We need to spend the next 2 days talking about the autonomic nervous system. We'll talk about the anatomy, we'll talk about the physiology, we'll talk about the pathways. We've got a fair amount to talk about. We've primed ourselves in some ways to make sense of this stuff, so hopefully it won't be too bad. And as I mentioned in the last class, some of the stuff we're going to be talking about is really some of the most important stuff you're learning in this class. The stuff that'll help you the most when we get to the next section in 431, because in 431, when we talk about all these autonomic processes of the cardiovascular and the respiratory and the digestive system, you'll be armed with the information of understanding how these things are controlled. We still have 2 more assignments due. One unfun one, and that is your unit reviews. I know you've had so much fun doing those, so we only have 1 more of those to do, and that's due tomorrow. Also, from the fun standpoint, you've got that reflex lab. So like I said, most of the materials that you need for that are things that you should have. If not, then I want to encourage you to, again, fill them out do them as a thought experiment. Don't just leave things blank. Please fill them out. Even if you guess wrong, it's better to guess than to just say, I couldn't do this or I don't know. So please do that properly. And there's a bunch of reflexes you'll be playing with on that, and that's due tomorrow as well. Hopefully you've got most of that done this weekend. And then Thursday and Friday's exams. Thursday, you know exactly what to expect. Lab and lecture exam. Complete them in either order that you want. Complete them within the time of the classroom. Probably not as much anatomy as in the last exam. Probably closer to 60 or 70 questions would be my guess on this one, but I haven't finished putting it together yet, so I don't know for certain. And then Friday is your final exam. Again, you have the whole class time to complete it within, but again, the exam, whenever you start it, you will only have 100 minutes. So you will have 100 minutes to complete 100 multiple choice questions. And again, that is the final exam. It is cumulative, covering everything we've covered, every chapter, every topic, everything we have covered in this class, you are responsible for on that. All
I
right?
got a question.
Yes.
In regards to the final exam, we have 100 minutes, 100 questions. Is there any way to get an extra 5 minutes so we can do that scanning the ID? Because what I saw is every time we start a test, the time starts before you even scan the ID. Sometimes I'm having trouble scanning it, and that takes like 2-3 minutes. And then, show the room in the beginning, show the room in the middle of the test. That takes another 2-3 minutes, and usually it takes me at least a minute to do a problem, so
yeah, I would consider adding 5 or 10 minutes to the final exam. I can look at doing that. That seems reasonable.
Okay, thank you.
Yep. Any other questions? All right, excellent. Well, then let's dive into the new material. All right, so again, our goal is to talk about the autonomic nervous system. As we've kind of already discussed in this class, the autonomic nervous system regulates the activity of what effectors? What effectors are controlled by the autonomic nervous system? There you go. Smooth muscle, cardiac muscle, and glands. Excellent. Perfect. Excellent. All right. So it regulates the activities of those things, right? Giving us that involuntary control. Oops. There you go. So we can go ahead and get rid of that. Smooth muscle, cardiac muscle, and glands. That involuntary control of those structures. So like we talked about, you don't have to, after you've had that cheeseburger for breakfast, tell your stomach to start contracting. You don't have to tell your salivary glands to start producing saliva, or your tear ducts in the morning to start doing those as well. It's all that involuntary control. And as we talked about in the last class, right, job of this is to maintain homeostasis. And again, it is outside of our conscious control, so it uses those reflex arcs that we talked about. where we have a receptor, we have an afferent pathway, we have some type of integration in the central nervous system, a motor pathway to an effector. So again, this is all stuff that we know and we have talked about. One of the things that I want to make sure that we understand, though, is that when we talk about the autonomic nervous system, and I want to write this big here so that we make sure we understand this. The autonomic nervous system is a motor branch. It is a motor pathway, branch, or we can say pathway. If you think about, for instance, that salivary gland that we were talking about, right? Obviously, when you put that food in your mouth, right, we get that, excuse me, that taste sensation, sweet, salty, umami, all those things we've talked about. So our special senses are involved in sending information to our brain to tell our salivary glands to get ready. Your stomach, when it stretches, as we talked about, that internal activity, that internal stimulus, so the stretch of the stomach sends a signal to tell the stomach to churn more, to produce more hydrochloric acid, right? Seeing, right? Again, there's nothing that looks more amazing than a Carl's Jr. Hamburger on their commercial. Again, the burgers themselves may not look that impressive, but on a commercial, just the look of that, or the smell of your neighbor's barbecue. I mean, there's all these different types of sensory, all types of sensory information is used to determine the activity of our smooth muscle, our cardiac muscle, and our glands. Right? So we use those interoreceptors, what's going on in the inside. We use our somatic receptors. We use our special receptors. All of that information is gathered to determine how we control that smooth muscle, how we control that cardiac muscle, how we control that glands. So when we talk about the autonomic nervous system, it is a motor pathway. It is a motor branch of the nervous system, right? And because it is something that we're not consciously thinking about, most, and again, of course, as always, that's the key word there, most is not all, but most of that control comes from nuclei. And remember, of course, a nuclei is a cluster of cell bodies in the central nervous system. Most of it occurs in the nuclei of the medulla and of the hypothalamus. And again, the key word there is most, because also, as we have also talked about, there can also be a processing that takes place in the spinal cord. And if processing takes place in the spinal cord, where would that autonomic nervous system processing take place? If only I knew where the autonomic motor neurons were located in my spinal cord. Autonomic in the anterior gray horn? Lateral gray horn, there you go. Excellent, so in the lateral gray horn. So you're right, gray horn, but lateral gray horn. Absolutely, so excellent. So that is where there is, and again, it's involuntary. These are reflexes outside of our conscious control. Now, the good news is this is a motor pathway. And we already have some familiarity with the motor pathway because we have talked about motor pathways in the somatic motor nervous system. And so one of the things that we're going to be able to do is compare our somatic motor pathways to our autonomic motor pathways to see how those work. There are some similarities in those pathways because they're both motor, they're both efferent, they're both going out, but they're going to be different in their effectors, they're going to be different in their pathways, and they're going to be different in the neurotransmitters that they use and the effects of those neurotransmitters. All right? So let's take a look at this. For this, I'm going to draw it first. And I want a line to separate them. Not sure why that line is orange, but doesn't really matter. There we go, that works. All right, so first let's talk about our somatic motor pathway. Oops. Now, like all of our pathways we're going to be talking about, there's some key components we need to discuss. We need to discuss where it starts in the central nervous system. So actually, let's make this a teeny bit smaller. There we go. Where it starts in the central nervous system. The pathway to the effector. Juggle some of these things around. Excellent. The pathway it's going to take to the effector. Obviously, the effector that we are going to have. And then the other thing we need to worry about is we need to worry about the neurotransmitter released and its effect. All right, excellent. In our somatic motor pathway, where does it begin? I'll give you a hint of where it begins. It starts somewhere in the spinal cord. Anterior gray horn, excellent. So absolutely, it starts in the anterior gray horn. We have a location. Excellent. Now that we have a location, you can tell me a little something about that neuron. That neuron that is located here in the anterior gray horn. What is its structural classification? Multipolar, excellent. We have three pieces of information that go together. Its function, somatic motor, its location, anterior gray horn, and its structural classification, multipolar. All right, excellent. So we have a multipolar neuron in the anterior gray horn. Excellent. Now, when that neuron leaves, but a little bit more specifically, when its axon leaves the spinal cord, what is the first thing that it forms? What does it enter? What structure does it become a part of? Ventral root, excellent. So it goes axon, travels out, the ventral root. And after it travels out the ventral root, what does it become a part of? What does it help to form then? Spinal nerve, excellent. Let's draw that. Let's add that anatomy. We know we have a dorsal root. Oh, no, don't want this to be purple. Undo black first. We know there is a dorsal root with a dorsal root ganglion, but we really honestly don't care about that because that's part of the sensory pathway. But what we do have is a ventral root that comes out and then becomes part of the spinal nerve. And so that's what our axon does. Our axon comes out the ventral root and becomes part of the spinal nerve, and it travels towards its destination. Now, there is one more piece of information we need to know about the somatic motor neuron, and that is that our somatic motor neuron's axon is myelinated. Of course, what type of cells would form that myelin on this axon? Schwann cells, exactly, because it's motor, it's in the peripheral nervous system, so indeed it would be Schwann cells that form myelin. And so let's cheat, and I'll just make little boxes. There's a box, and there's a box, and there's a box, and there, and there, and there. So you get the idea. Excellent. And I'll finish my myelin in just a second. But let's jump ahead. What effector is our somatic motor pathway going to terminate at? What does our somatic motor pathway communicate to? What type of tissue does it terminate at? I believe that's the exact question that we had on your review, right? Excellent. Skeletal muscle. Careful, not just skeletal, because we want to make sure we're not talking about bone. We need to talk about skeletal muscle. Excellent. So we know it's going to be skeletal muscle. And let's again go ahead and cheat and draw ourselves a cell here. A striated tubular cylinder shape. I know these are all supposed to be parallel, but you get the idea. And heck, let's throw a couple nuclei on the periphery as well. Excellent. We know this somatic motor neuron is going to travel all the way to our skeletal muscle, where we have our neuromuscular junction. And again, let's go ahead and myelinate this the whole way. And then, of course, what neurotransmitter do we know this somatic motor neuron is going to release? Acetylcholine, excellent. It releases acetylcholine. And what is the effect of the acetylcholine? Is it excitatory or is it inhibitory? Yeah, it's always excitatory. Excellent. So it's going to release a little bit of acetylcholine here. And we know that the effect of that acetylcholine is, oops, I want that to be purple, is going to be excitatory. Excellent. Notice we really didn't need the drawing. I like it because it helps me to visualize, and I enjoy drawing. But in words, we have done a good job of describing this somatic motor pathway. We start with our somatic motor neuron, which is a multipolar neuron in the anterior gray horn. That somatic motor neuron's myelinated axon travels out the ventral root through the spinal nerve, where ultimately it is going to terminate at our skeletal muscle, always releasing acetylcholine and always excitatory. And if you think about it, with the exception of the fact that I've told you specifically that this axon is myelinated, even though we've said most axons are myelinated, there really isn't a lot of new information here, which is convenient because we don't like new information. So again, in many ways, this is just kind of a bit of a review. All right, questions on that? And hopefully there aren't too many because like I said, this isn't new information, but if this makes sense, then this will help us to make sense of the rest of this. All right, stunned silence always lets me know that I understand this entirely. Excellent. Let's move forward. So now that we have talked about what we know with our somatic motor pathway, let's talk about our autonomic. There you go. That's right. Looked weird. Now, we are going to still worry about the same types of things. We need to know where it starts in the central nervous system. We need to know our pathway to our effector. We need to know the neurotransmitter released and the effect of the neurotransmitter. And we need to know the effectors. Excellent. All right, let's start easy. What are the effectors again? I know we've already said it like twice, I think, already in this lecture, first 20 minutes, but let's beat the dead horse. What are the effectors in the autonomic pathway, motor pathway? There you go, excellent. Smooth muscle, cardiac muscle, and glands. Perfect, excellent. So since we like drawing, let's go ahead and draw some of these. I will cheat and use my diamond for my, oops, no, hold on, don't want that to be black. There's my spindle-shaped smooth muscle cell. I'll give it a nice big nucleus in the center. Now let's make some goblet shape glandular cells. There you go, and then we'll draw our branching. Striated, single nuclei, cardiac muscle cell. Excellent. So that was easy. All right, let's start with something else that should be relatively easy. Where it starts in the central nervous system. Where in our spinal cord are we going to want to start in our central nervous system? Where does our autonomic motor pathway begin? Lateral gray horn, excellent. Now I am going to have to put that pesky word mostly in parentheses, but for right now, we'll focus there, absolutely. And we know this is an autonomic motor neuron. We know its functional classification. We know its location in the lateral gray horn. That tells us we know one more thing about it, and that is, of course, its structural classification. And what is the structural classification of all of them? What is multipolar? Excellent. So all of these neurons are multipolar. So we are going to have, and let's start with red. A multipolar neuron whose cell body is located in the anterior gray horn. Its axon is going to need to leave the spinal cord. And of course, we know you have two routes out, the dorsal root with the dorsal root ganglion and the ventral root. And being part of the motor pathway, which way is it going to want to go? Dorsal root or ventral root? Ventral root, excellent. It is motor, so it's going to come out the ventral root to the spinal nerve. So that's going to be our starting point. Put that up here. Put this here. Dorsal, oh, not dorsal, ventral root. Spinal nerve. And then it continues on from there. We know one other important piece of information about this neuron, in that its axons are myelinated. So again, similar to what we see in our somatic motor pathway. Oops, that's horrible. All right. But that's where the similarity ends. Because what ends up happening in this pathway to the effector, while up here in our somatic, there is only one motor neuron. Here in our pathway to the effector in the autonomic, we actually have two motor neurons. So what that means is that this neuron actually is going to synapse on a second neuron. And it is that second neuron that is then going to carry the information to the effectors. Let's shorten that a little bit. That's not shortening it. There you go. And will then actually communicate to the effectors. So we have two neurons in this pathway. Now, if that's going to be the case, then we are going to need to worry about naming these two neurons so we can tell them apart. This first neuron, well, let's take it a step further before we identify these. Now, we know this cell body of the first one is located in the lateral gray horn. This second neuron's cell body isn't just going to hang out by itself. It is going to be clustered together with dozens, if not hundreds of others. And what is the term we give for a cluster of cell bodies that are in the peripheral nervous system? A ganglion, absolutely. This is our autonomic ganglion. Oops, and I want that to be black. And I want that to be big. Excellent. We have an autonomic ganglion here, where our second neuron is located. The reason I wanted to mention that first is because that's going to help us to understand the name of this first one. This first neuron that starts in the central nervous system and goes to the ganglion is called the preganglionic neuron. And we know several things about this preganglionic neuron. It starts in the central nervous system. It terminates, or synapses, if you like that term better. Let's do that. Synapses at the ganglion. As we said, its axon is myelinated. And we know two other things about it. First is that they always release and acetylcholine. And the other thing we know is that it always is excitatory. So this one, again, let's be clever here, there. So we're releasing acetylcholine there as a reminder for that. And we're releasing acetylcholine here. Oops. And as we mentioned, this is always excitatory. All righty.
Professor,
Yes.
did you say that it starts in the synapses at the ganglion?
No, it starts in the central nervous system.
Oh, okay.
Right. In this case, in the lateral gray horn of the spinal cord, and it synapses at the ganglia. So where it ends. So it starts and ends, or synapses
Gotcha.
or terminates, whatever term you like best for that. Right. So again, for that we could say, it ends, terminates, whatever word makes sense to you. That's how we would describe it. So that's where it ends or it terminates at that. All right. Let's cheat. Oops. No. Okay. That can go there. That's what I want. Move that over here a little bit more. All right. Any other questions on the preganglionic neuron? Excellent. Now, if the one that starts in the central nervous system and terminates at the ganglion is the preganglionic neuron, well, then guess what we call the one that starts in the ganglion and terminates at the effector? There you go, jumping ahead. Postganglionic neuron is the second neuron in this pathway. Again, this one starts, and again, let's be specific. When I say starts for both of these, what I really mean is, where is the cell body? Right? That's really what I mean when I say starts. And of course, it starts in the ganglion. This one synapses or ends or terminates at the effector. But there's another couple differences other than the anatomy of them. Here, this postganglionic neuron's axon is actually unmyelinated. The other big difference is in the neurotransmitters. Some postganglionic neurons also release acetylcholine, where others release a different neurotransmitter called norepinephrine. And is there another fancy name for norepinephrine? Well, is there another name for epinephrine? What's the other name for epinephrine? Adrenaline. So this would also be known as noradrenaline. Either of those are acceptable terms. So again, some will release. Oops, no. You get to stay at 14. You get to be smaller. So some release acetylcholine and some release. And once you've written out norepinephrine once, you can abbreviate it capital N, capital E. Now, again, let's be clear. This one specific postganglionic neuron that I've drawn, on Mondays, does it release acetylcholine, and on Tuesdays, it releases norepinephrine? No, of course not, right? There are millions of postganglionic neurons in your body, and each one, right, some will release acetylcholine and always release acetylcholine, some will release norepinephrine and always release norepinephrine, okay? And it's the same thing with the effects. When we talk about the effects, as we know, some synapses will be excitatory and some synapses will be inhibitory. So when we talk about effects, again, some will be excitatory and others will be inhibitory. It's going to depend on the synapse, depend on the effector, right? Let's think of our fight and flight response. When we have a fight or flight response, what kind of effect do we want to have on our heart rate? Do we want our heart rate to go up or do we want our heart rate to go down?
Go up.
All right, so there we want it to increase. However, when you're stressed, like say for instance, you're in the classroom and you need to stand up and give a 20-minute presentation. When you stand up to give that 20-minute presentation, right, your heart may be beating really fast in your chest, but how much saliva are you producing in your mouth at that period of time? What about saliva production in that fight or flight? Do you start foaming at the mouth, salivating when you get stressed?
You probably
So there, it's going to inhibit production of saliva, right? That's why you get dry mouth when you get nervous. So again, in some cases, you'll see excitation. In other cases, you'll see inhibition. All right. So again, notice we described this all in words, which is what's going to be expected of you. But I like the pretty pictures because I think sometimes it helps to make sense of this.
I got a question.
Yes.
For the autonomic motor pathway, they both start in the lateral part, correct? In the gray horn, lateral gray horn.
But what do you mean by both?
You said he has two, correct?
Two neurons, yes.
Yeah. So they both start in the lateral gray horn?
No. Only the preganglionic neuron starts in the lateral gray horn. The postganglionic neuron starts in the autonomic ganglion.
Oh, okay. Okay.
Yeah. Again, this is remember, these are pathways, right? So in the somatic motor pathway, one neuron leaves the house and carries the signal all the way to the effector. Right? In the autonomic pathway, it takes two people to send that message. First person leaves the house, gets halfway, gives it to its relay member, and then the relay member takes it the rest of the way to the effector. Right? So if you're running a relay race, not everybody starts at the start, right? Just the first person does, and then the next one picks it up along the way. All right? Again, I've like drawing this way, but again, your book's got pretty pictures that shows this very, very nicely as well. Again, we have described our somatic motor pathway, one neuron with a myelinated axon, starting in the anterior gray horn of our spinal cord, going all the way to skeletal muscle, releasing acetylcholine where it's always excitatory. And here's the pretty picture from the textbook that shows that. The effector is skeletal muscle. Again, this is what gives us our voluntary control. It always releases acetylcholine. It is always excitatory. And as we know, it starts in the anterior gray horn, ventral root, spinal nerve, to our destination. What's new that we learned about today is our autonomic motor. Now, as we already knew, the effectors are going to be cardiac muscle, smooth muscle, and glands. But then we've added some new information. Autonomic motor neurons have two different neurotransmitters they could potentially release, either acetylcholine or norepinephrine. And in fact, that norepinephrine in our sympathetic pathway, that adrenaline, can be released into our blood to give us that big, huge global effect. And their relationship with the effectors is more complicated. Some effectors are going to be excited, some effectors are going to be inhibited. But the biggest difference is in the anatomy. To get from our central nervous system to our effector in our autonomic nervous system requires two neurons. A preganglionic neuron, whose cell body is located in the central nervous system, specifically in the lateral gray horn. It's multipolar, right? And just like this one is multipolar as well. I guess we didn't emphasize that. I don't remember if I emphasized that. Let's go back to the picture and see. Did I emphasize that? Nope, I did not. So let's emphasize that for both of them. And what color haven't I used yet? Let's go orange. These are both motor neurons. Oh, don't need to be that big though. These are both motor neurons, so both are multipolar. Oops. I mentioned that it was multipolar here for the first one we talked about in the lateral gray horn, but it is important to remember that both of these are motor neurons. So both of our neurons are multipolar. So let's go ahead and draw some dendrites on this one. Excellent. There you go. Now I'll take my picture again. So we have that. So two multipolar neurons. The preganglionic neuron cell body is located in the lateral gray horn. Its myelinated axon leaves the spinal cord and synapses at the ganglion, where it always releases acetylcholine. And it is always excitatory. Our preganglionic neuron cell body is located in the ganglion. It is also multipolar. Its axon, which is unmyelinated, travels to our effectors, where it can release acetylcholine or norepinephrine, right, one or the other. And the effects, some will be excitatory, and others will be, inhibitory. And again, it's not, like I said, it doesn't switch. It's not like on Monday when you get scared, your heart beats faster, and on Tuesday when when you get scared, your heart beats slower. What I mean is that when you get scared, your heart beats faster, but when you get scared, your salivary glands decrease in activity. So some effectors are going to be excited, and some effectors are going to be inhibited. All right. Questions on that? All right, excellent. Your book's got another nice, pretty picture illustration that shows this as well. Again, notice here we have our cell bodies starting in the central nervous system. Here we have our effectors. Here is our somatic motor pathway. One neuron from central nervous system to effector. myelinated axon, releases acetylcholine, always excitatory. Here in our autonomic nervous system, we have cell bodies starting in the central nervous system, but we have ganglia in between. So it takes 1, 2 neurons to get to our effectors. Some release norepinephrine, some release acetylcholine, some are excitatory, some are inhibitory. And as we also hinted at, we also have the ability to release these neurotransmitters into our blood so that we get that big global effect too as part of our stress response. And that's really the next step in this. We have talked about the difference between the autonomic nervous system and the somatic, but this picture shows us one other thing as well. As we know, there are 3 subdivisions, 3 branches of our autonomic nervous system, the sympathetic and the parasympathetic. And as we'll learn, those pathways are actually going to be slightly different from each other as well. So we're going to have to compare them and their pathways as well. All right, questions on that? All right, if we're comfortable with the difference between our somatic and our autonomic, then let's focus now just on our autonomic. In our autonomic nervous system, as we just finished mentioning, there are 2 subdivisions, our sympathetic and our parasympathetic. And as I mentioned, there are some cutesy mnemonics that we use for these. What's the cutesy mnemonic we use for the sympathetic? There you go, fight or flight. And as I mentioned, that's actually a pretty decent mnemonic, because really what our sympathetic nervous system is involved with is with dealing with stress. Or more specifically, mobilizing energy to deal with stressful situations. To deal with those stresses that come at us. All right. We also had a cutesy mnemonic for the parasympathetic. What was the cutesy mnemonic for the parasympathetic? Rest and digest. That one's not quite as relevant as the fight and flight. We'll stick with it for right now, but as we'll see, there are actually some other mnemonics that people use that are more accurate, and there's a little bit more involved with this. Our sympathetic is kind of our housekeeping processes. Or more specifically, not mobilize energy, but the storage of energy, or the maintenance of energy. And so we have these 2 branches of the autonomic nervous system. And one of the important things, and I'll make a point of emphasizing this before, but as you can see, they're very different in their functions. And in fact, more specifically, they are always antagonistic of each other.
What does that mean?
I was going to explain that in just a second. So in our heart, for instance, when you have, oops, I spell heart right. When you have a fight or flight situation, what does our sympathetic nervous system do to our heart rate again?
Makes it go up?
Yeah, it makes it go up, absolutely. So it excites, oops, wait, hold on. It increases it, excellent. So it excites the heart, increases heart rate, increases blood pressure, things along those lines. When you've got that rest and digest, or when you're doing those housekeeping or storage, or when you are meditating, right? When you're doing those types of activities, right, that is stimulating our parasympathetic nervous system. And what happens to your heart rate? Decreases it, or inhibits. Now, doesn't mean that our sympathetic increases everything and our parasympathetic decreases everything. So let's take something else instead, stomach. When that bear with an axe walks in the room and you have to decide whether you are going to fight it or outrun someone in the classroom with you, is your stomach so much concerned about, is our sympathetic nervous system so much concerned about breaking down that cheeseburger you had for breakfast? No. So it's going to inhibit activity or decreases. However, when you're doing that, it's in the name, rest and digest, when you're 3 plates into Thanksgiving, what kind of effect is your parasympathetic nervous system having on your stomach?
Increase.
Yeah. So antagonistic means they always have opposite effects. If one excites, the other inhibits. If one inhibits, the other excites. They're always going to be opposite in their effects. And if you think about it, this is what allows us precision of control. Take it outside of the realm of the body and inside of the realm of your household, right? As we've talked about, especially now that you have these smart thermostats, you can set the house to a specific temperature and maintains it because you have 2 antagonistic forces. You have your air conditioner, which brings things down, and your heater, which brings things up. And that is what allows you to provide precise control of the temperature of your house, right? Right now, at the end of July, if you only had an air conditioner, you could control the temperature of your house. Because as it gets to start to be 90 and 100 outside, your AC can come on and bring it back down to 78. And then it goes up and the air conditioner brings it back down, and it goes up and the air conditioner brings it back down. And you're fine until we reach about October. And October, when it's 56 degrees outside, is that AC going to be doing you any good to help you to control the temperature in your house? No. You need that heater to be able to bring it up. So having those opposite effects, those antagonistic inputs, allow us to maintain very precise control of the functions of our organs. So again, we have this dual innervation to most effectors. And of course, dual innervation means that they receive input from both the sympathetic and parasympathetic. And again, as we talked about, that allows for precise control. Of course, there is one key word in this sentence. And what is that one key word in that sentence?
Most.
Most. That means not all. There are a few effectors in the body that only receive input from the sympathetic nervous system, that don't have that dual innervation. And we'll identify what they are in just a moment. So there are a few exceptions. And again, the advantage of that is that we get this constant input. Remember, one of the things that we talked about when we first start talking about cardiac muscle is I mentioned to you that if I was able to rip your heart out of your chest and put it in a beaker and provide it with all the oxygen and all the nutrients that it needed, it would beat. And not only would it beat, but as I mentioned, your heart rate of your heart would be about 100 beats per minute. Now, while it's sitting inside of your chest right now, is it beating 100 beats per minute? Right, unless you hit Dutch Brothers for a quad shot on the way here, hopefully your heart is not beating 100 beats per minute. So even while you're sitting here at quote unquote at rest, there is input to your heart from the sympathetic and the parasympathetic, and more sympathetic than parasympathetic, which is slowing your heart rate down lower than it actually wants to be right now. So that autonomic tone, you constantly get an input from both sympathetic and parasympathetic. Now, is it always equal amounts of both? No. Sometimes there's more sympathetic, sometimes there's more parasympathetic, but you're usually getting input from both. But like I mentioned, there are some exceptions that only receive input from the sympathetic nervous system. Quad shot is the only way you can wake up. Yeah, for some people, unfortunately, that has become the case. Absolutely. Now, there is, as I mentioned, some exceptions. And the good news is most of them make sense on the surface. Obvious stuff that kind of makes sense on the surface, but they all make sense once we have a chance to talk about them. So one of the most obvious organs that only receives input from the sympathetic nervous system is the adrenal gland. What, of course, does the adrenal gland do? Not a trick question. Yeah, makes and releases adrenaline, right? And when do you want to release adrenaline into your body?
Fight mode.
Yeah, when you're in that fight or flight situation, when you're in that stressful situation, absolutely. Right? That adrenal gland releases that adrenaline to help us to deal with that stressful situation. Excellent, right? So again, notice we don't need, yeah, exactly. When that bear with an axe walks in the room, your adrenal gland needs to release adrenaline. So while you're sitting here calmly just listening to me talk, right, you shouldn't need any adrenaline, so there's no reason for the parasympathetic to communicate to it. Some of the other obvious structures that only receive innervation from the sympathetic is the skin. The skin only receives innervation from the sympathetic nervous system. So that means things like your sweat glands, things like your erector pili muscles, right? All of those are things that only, whoops, hold on, no, wrong direction. I want that, that's what I want, there we go. So again, when do you get sweaty? When you're vigorously exercising, right? When you're using energy, mobilizing those energy for resources, right? When does your hair stick up on end, right? When you're cold, so you're using energy to try to shiver and bring your body temperature up. When you're scared is when your hair stand up on end. So again, your skin only receives input from the sympathetic nervous system, including all of the blood vessels going to your skin. But not just to your skin, but all the blood vessels going to most parts of the body. There's that damn pesky word most again, though. All of the blood vessels in your body receive input from the sympathetic nervous system and the sympathetic nervous system only, except for one big exception. There is one set of blood vessels that receive input from the parasympathetic nervous system. And you may not have thought of it in those terms, but I know you're aware of it. Because again, back in ancient times, there were things called dates, where you would get to find somebody attractive. And when you found that somebody attractive, you could take them outside to a nice fancy restaurant or something along those lines. And like you, like me, right, there's always that big important third date. When you take your man out for that big important third date, right, usually you take your man out to a really nice restaurant, buy him a really nice meal, right, maybe get him a nice gift, because there's an expectation on that third date. And that man knows that. He's well aware that you're taking him out on that third date, and there are some expectations of that. And that can be a bit of a concern, because if your boy is a little bit nervous, does that nervousness help performance? No, right? He may have a difficulty rising to the occasion if he is too nervous, right? So of course you do what I always do on my third date, ply him with a little bit of alcohol, and then he'll be a little bit more relaxed, and you can get what you need from him, right? Now, of course, the reason for this is the blood vessels that control your erectile tissue. That means obviously the penis in the male. That means the clitoris and the vulva in the females. Those blood vessels that control the erectile tissue are actually controlled by the parasympathetic nervous system. When we get to the reproductive system in 431, and we'll talk about this a little bit more, as we'll see, as we'll talk about then, arousal is a parasympathetic reflex, whereas orgasm is actually a sympathetic reflex. So a cute mnemonic that you can use to remember that when we get to the reproductive system in 431 is point and shoot. Right? The pointing, the arousal is a parasympathetic, P for point, P for parasympathetic. Shoot, the orgasm, is a sympathetic, S for sympathetic, S for shoot. So if you can remember point and shoot, you can remember arousal and orgasm, and you can remember that the one set of blood vessels that is innervated by the parasympathetic nervous system are those that control the erectile tissue. All righty? Questions on that? All right, excellent. So again, I think most of these make sense, right? When we think about these types of effects. The one that I think on the surface doesn't make sense is this last one, the kidneys. What is it that the kidneys do for us?
Is that the one that filters the blood?
Yeah, absolutely. Filters the blood, absolutely. Filters the blood. And it does it a lot. While you're sitting here at rest in lecture like I am, calmly in this class, not moving around a whole heck of a lot, about 25% of your blood is being directed towards your kidneys. During the course of the day, your kidneys end up producing over 200 liters of filtrate. Now, do you produce 200 liters of urine during the course of a day? Anyone? Hopefully not. If you are, you're probably listening to this lecture in the bathroom, right? No, we produce about 1 to 2 liters of urine during the course of the day. So we filter that blood. Most of that filtration goes back into the blood. And filtering the blood is vitally important. How long can you survive with 1 kidney? Anyone? Everyone knows we have 2 kidneys. How long could you survive with 1? Absolutely. Your whole entire life. Absolutely. Right? One of the things we've learned is life is lazy. But in our kidneys, we have a huge redundancy. Right? You could injure a kidney. Right? You could lose a kidney. You could sell a kidney on eBay. And you'd be able to still live your whole entire life because 1 kidney is capable of doing all of the filtration that is necessary. Right? But what happens if you sell 1 kidney on eBay and make a lot of money, so much money that you think it's a good idea and you decide to sell your second one? How long can you survive with no kidneys?
Yeah.
Yeah, not very long. Hours, maybe days, right? Dialysis is a short-term solution, but it's not going to allow you to survive long-term, right? You need that kidney. Being able to filter our blood is a vitally important process. Now, does filtering the blood sound like stress, or does filtering the blood sound like housekeeping?
Housekeeping.
Housekeeping. So you would think that the kidneys, therefore, would be controlled by the parasympathetic nervous system. But it turns out that filtering the blood is such a vital process that we don't need our parasympathetic nervous system to tell it to do that. Here's where we run into the problem. Remember, as I mentioned, while you're sitting here calmly in your chair, 25% of your blood is going to your kidneys. So your kidneys have a huge blood volume to them, which gives them a huge blood pressure, and they use that to do all of the work. Great question, Ryan. My guess is that no, you would not produce quite as much filtrate, but my guess is that it would be close. Because again, we are able to do enough to survive from it, and not just survive, but thrive. You wouldn't see any impairment of function. The one morphological difference that I do know that occurs that is when someone gives up a kidney is that their remaining kidney will increase in size. So the kidney will get larger in size. But my guess is that, so it's probably able to get close to it. My guess is that it's not going to be able to function exactly the same as one. So you probably see a decrease, but my guess is that it would go up. And so it would probably be close. Yeah, maybe something like that. Cool. Wow.
Well, that if you have just one, isn't that gonna affect like if you're a heavy drinker?
It can. Drinking has more of an effect on the liver than it does on the kidney, but sure, it would affect the kidney as well. Yeah. So yeah, it's probably not recommended. But you know what? Even if you have two kidneys, you're not used to be a heavy drinker. So, all right. So again, it definitely sounds like housekeeping, but it turns out it's such a vital process, we don't really need the parasympathetic nervous system to tell it what it does. As I mentioned, while you're sitting here in class, sitting at your chair, listening to me talk, 25% of your blood is going to your kidneys so that your blood pressure is high and that blood volume in the kidney is high, and so it is able to function at its maximal efficiency. However, then we take a 15-minute break and you go running to the kitchen. Or better yet, it was when we were at school, you'd go running over to Starbucks to get some coffee and come running back. When you get up and briskly walk from here to Starbucks, as you're briskly walking from here to Starbucks, is 25% of your blood still going to your kidneys? No, it's going to be going to your muscles. It's going to be going to your lungs. It's going to be going to your heart. So what happens is when active, the amount of blood going to the kidneys decreases. And if there's less blood going to the kidneys, we have less pressure, we have less volume, and we could see a decrease in function. This could cause a decrease in function. And as we talked about, filtering our blood is vitally important. So every time we get up and walk around or we carry a bag of groceries up the flight of stairs, we don't want our kidney function to go down. So when active, the sympathetic nervous system stimulates the kidneys to maintain optimal functioning. So if you're if you're briskly walking from here to Starbucks, right, or taking your dog for a walk, which is what everybody's doing nowadays. Everybody's dogs are probably the most fit they've ever been over the past few months because everybody's getting out and walking with their dogs. Every time you do that, we don't want kidney function to go down. So our sympathetic nervous system is going to help to compensate for that by stimulating the kidneys. Now, are there limits? Absolutely. If instead of walking my dog, I am sprinting, if I am maximally intensely working out my body or fighting that bear with an axe, dealing with some major stress, is there going to be a decrease in kidney function during those times? Yes. But as we talked about, how long can you sprint for? How long is a fight with a bear going to take? So yes, in maximal intensity stressful situations, there can be a decrease in kidney function as a result of that. And that's one of the problems that we keep talking about. This sympathetic stress responses we'll talk about helps us to deal with life or death situations, because from an evolutionary standpoint, those were the stressors, right? Bears with axes roamed the countryside, right? And as a result of that, we needed to be prepared. When we were stressed, it was usually a life and death situation, right? Now it's not a life and death situation. The types of things that stress us are, viruses that we can't see, or whether or not we're going to have a job next week, whether or not you have a 10-page paper due, all of these types of things. And those types of stresses, that chronic stress, does it have the potential to then have an effect on kidney function? Yeah, it can. No, great question. So, and again, obviously, we'll talk about this in great detail when we get to the urinary system in 431. But the short version is that what happens is, well, sort of, uh, the blood is filtered out, fluid, small solutes are filtered out actually into tubules that are contained within our kidneys. Our kidneys have almost a mile's worth of these tiny little tubules where we basically pull out the fluid and then filter it back and forth. And what happens is everything that gets brought back into the blood supply stays in the body. Anything that stays in that tubule eventually becomes part of your urine and leaves the body. So it never quite makes it into the interstitial fluid. There are tubes that collect it. And then either it's brought back into the body or it leaves in our urine. All right. So like I said, I think on the surface, because again, we think of our kidneys as a housekeeping function, it doesn't make sense. But when we understand that when we're active, less blood goes to the kidney, and we still want the kidney to be able to maximally be efficient, it kind of makes sense why the sympathetic would stimulate the kidneys. All right. Questions on that? Oh, I see what you're saying. Yes. Yes, basically, absolutely correct. The function of the cardiovascular system is to move solutes throughout our body. When it gets to the capillaries, those are then exchanged with the interstitial fluid. So basically, whatever's in the interstitial fluid is going to end up in the blood. Whatever's in the blood is going to end up in the interstitial fluid. Absolutely, they intermix, they interchange with each other, and essentially, interstitial fluid is filtered blood plasma. So yeah, absolutely. All right, excellent. So those are our two divisions. We need to talk about them both physiologically and structurally. I think the physiological makes more sense because, again, when we think fight and flight, we all have an idea of what those mean. So I think we'll talk about the physiology of these first, and then the anatomy is where things get a little more convoluted. But I think this is a good stopping point for our first break. It is 9:12, so let's come back at 9:27. And at that point, we will talk about all of our physiological effects of the autonomic nervous system, and then we will dip our toe into the anatomy. All right. So, like I said, we will restart at 9:27, and I will start the recording at that point. All right. Any other questions before we take our first break? All right. See you guys in 15 minutes. So, like I said, I want to focus on the physiological effects of the autonomic nervous system. So let's start first with the fight or flight. Give me some examples of when you're in that stressful fight or flight situation, some of the physiological changes that occur in your body.
Besides, like your heart beating fast, maybe sweating.
Yeah, well, so all of those are great examples. So heart rate increases. And not only does the heart rate, but also the strength of the contractions. Right, you can feel it pounding in your chest. Heart rate increases and strength of contraction. Increases. Excellent. I like all of those. Hold on a second. You guys are getting ahead of me. So I love all these. So, okay, that's a perfect one. So then also increased breathing, increased respiration, and dilation of the airways. Right, I think I've already mentioned in this class that my daughter has an extreme peanut and tree nut allergy. So as we've talked about, everywhere we go, we bring an EpiPen with us. Why do we bring epinephrine, adrenaline with us everywhere we go? Because one of the things that could happen if she gets, if she comes in contact with one of these allergens, is she can go into anaphylactic shock. Basically, two main events occur. What happens is you get a rapid dilation of your blood vessels, and that causes your blood pressure to drop. So you have difficulty getting blood up to your brain, which means no oxygen to your brain, which can be a bad thing. The other thing that can happen is it can cause an increase in mucus production, causing the mucous membranes of her airways to constrict, making it hard for her to get oxygen. If she can't get breath in, she can't get oxygen to her brain if there's no blood pressure. So what do? You stab her with that EpiPen. It increases heart rate, increases the contractions of the heart, increases blood pressure, right, as a result of that. And that increase in blood pressure can help to bring the blood back to her heart. And it dilates, and let's be specific, it dilates the smooth muscle of the airways to try to get more air in. Excellent. All right. Those are some other good things. It decreases digestive function because digesting food requires a lot of energy, and we don't want to waste food that way. But the other thing, as I mentioned, is that it is going to release energy or release glucose and triglycerides into the bloodstream. Oops. All right, from the liver, so it causes the liver to release, and from adipose tissue. Right, because again, fighting that bear with an axe, running from that bear with an axe requires energy. Remember, this is a fight or flight stress response. And like I talked about, that's one of the problems. This is an evolutionarily ingrained into our bodies. This is our bodies respond to stress. The problems are the things that stress us now are not the same as they were before. Right? The things that stress us now are our jobs, our life, our kids, right? That 10-page paper you have due on Friday, that lab and lecture exam you have on Thursday. Those are the types of things that stress us. And unfortunately, the way we deal with those stresses is by doing what I'm sitting here, sitting on the computer and taking notes or writing things out or reading a book or doing those things before. I think as I've mentioned in this class, I was glorious before I had kids. Right? But the problem is all this stress releases all this glucose into your blood. And if you don't physically use it, then it gets stored as fat, right? As a result of that. Excellent. So what else? Those are all good things. Oh, I think someone said sweating and sweat production. What else? Excellent. Pupils dilate. Right? We want to let more light in. Bears with axes are tricky. They can blend in with their environment. So being able to dilate your eyes allows more light to get in. I wouldn't say necessarily that your senses get more acute. Like I said, your pupils definitely dilate to let more light in. And so with more light coming in, there's more information to be processed. You may pay more attention. So really, I think what that response is you're going to pay more attention to those potential stimuli in that type of situation, right? You're paying more attention to those sounds and noises. You got to remember, we are inundated with a fire hose of information that is coming into us again. So we didn't really talk about what the intention structure in the brain is. There are a couple different areas that allow us to focus. There isn't just one that moves our focal point around because there's all sorts of different types of information that we're receiving. We ignore most of the information. Again, like the example that I love to use right now is the feel of the socks on your feet, the feel of the chair on your butt. That is information that is constantly coming into your brain. Hey, we're sitting on something. Hey, I feel something with my back. I feel something with my butt. But most of the time, we're not paying any attention to that, or the feel of the socks on your feet. You don't pay any attention to that. Now, if someone like me points it out to you, then you go, oh, yeah, I feel those socks on my feet. I feel that chair on my butt. That information is always coming in, but we're not necessarily paying attention to it. So the reticular formation is the one that affects alertness, absolutely. So the influence on alertness was the reticular formation. So yeah, so that was that one. All right. So yeah, so we may pay more attention to outside stimuli, but it isn't that our senses get better, although our pupil does dilate to let more light in. Anything else?
What about release of adrenaline?
Excellent. So one of the things that's going to happen is we are going to affect glandular activity. Right. So you're right. One of the things that we are going to do is we're going to release adrenaline. Why? Why do we release adrenaline? What's the advantage of adrenaline? True. Adrenaline does do most of these effects. So what's the difference between adrenaline and our autonomic nervous system? Yeah, one of
Is
them,
it
absolutely. We get a big global spread because adrenaline is released into the blood. And once a chemical signal gets into the blood, where can it go? Everywhere, right? So absolutely, we get a global spread because after all, we don't want our heart beating faster. And then 3 minutes after that, our eyes dilate. And 5 minutes after that, our airways dilate. And 3 minutes after that, my glands, my salivary glands decrease in their activity, things along those lines. We want it all happen at once. The other advantage of adrenaline is it's a hormone. And hormones, remember, are longer lasting chemical signals. Right? I will spare you right now, but usually this would be the point of time where I would scream really loud into the microphone to scare you, right? To jump. Or I'll use the example of late at night, the house is all quiet, you're sitting in there, and bang, there's that massive, car backfiring outside. And so you hear this big, huge bang outside, and you jump and you're startled and you're scared by that. Right? But almost immediately you realize that that was just a car backfiring, right? And not, Trump's Gestapo coming in to try to arrest you or anything along those lines. So you know you're okay that way. But does your heart rate instantly go back to normal after you've been scared? No, because you release that adrenaline. That adrenaline keeps and maintains that elevated stress situation for a longer period of time.
question?
Yes.
Does some of your pain receptors decline during a fight mode? Like, for example, like if you're just hanging around with your friend and your friend hits you or slaps you, you kind of like, make a big deal out of it. But when you're in a fight and you get like a bunch of punches and slaps, sometimes you don't even feel them until the fight is over or something like that.
So again, I think that so there are two things involved in that process. Part of the things, and again, we'll talk about this more when we get to adrenaline and some of our other stress hormones. Adrenaline isn't our only stress hormone. Corticosteroids are another example of, or cortisols are another example of a stress hormone that is released. Those can suppress your immune system. And decrease inflammation and things along those lines, so you can be less affected that way. But again, a lot of these things that you guys are talking about, and they're all absolutely correct, but really they're top-down processing, right? That injury is much less important to you when you are in that fight or flight, death fight situation, as opposed to if you're just sitting there and suddenly got hit out of the blue, right? And you're not in that stressful situation. So a lot of it has to do with your motivation. Like we talked about, me as that adolescent having to hold up that textbook, right? I tried to hold it up for as long as I could. I wouldn't, and I got swatted. But as I mentioned, if instead I dropped that textbook, I was going to get shot in the head, then based on that motivation, I would have been able to hold that book up longer. Not necessarily because being stressed by that made me stronger. It was just my motivation allowed me to push my body further than I necessarily needed to do. So it was really, I think a lot of that has to do with motivation, right? And paying that attention. So those types of things, I think would be effective in there. All right. The other thing that we're missing from this list is a redirection. I like saying it that way, of our blood supply. You have somewhere around 6 liters of blood in your body. And is that enough for every single part of your body to get all the blood it needs for maximal intensity of effort at all times in your body? No. So we need to be able to redirect blood to where we need it in these stressful situations. What that means is in some areas, we're going to dilate blood vessels. Of course, if we dilate blood vessels, that is going to bring more blood to those areas. And what are some of the locations or structures in the body that we would want to increase blood flow to?
Muscles.
Excellent. To our muscles. Where else? To the heart. Where else?
What about the lungs?
Absolutely, to the lungs to get oxygen. And by the same token, oops. And also, great question. No, as great as that would be, muscles, heart, lungs, liver, and adipose. Right? Because remember, we need to get the oxygen from the lungs. We need to get the sugar from our liver and adipose, get it to our muscles so that we can deal with that fight or flight situation. When you are stressed, do you always necessarily make the best decisions? I know there are a lot of students who think, no, when I'm in that last 11th hour and I'm having to write that paper, I do my best work when I'm stressed. Is that paper that you write in the 3 hours before it is due really better than the paper you would have written it if you had taken 2 weeks to work on it and research it and write it properly? No, because again, when you're in that fight or flight situation, the decisions are pretty simple. They're pretty basic. When a bear with an axe walks in the room, right, you don't want to have the big, deep philosophical discussion as to whether or not you should marry Sally, right? Or whether or not you should, take an extra semester to go abroad or something along those lines. You're not making big, huge, complicated decisions when you're in a fight or flight situation, right? It is run or it is fight, right? It is duck down or hide or whatever, right? So we actually don't get an increase in blood flow to the brain during those times. All right. And at the same time, we want to constrict blood vessels to areas that, bring less blood. To those areas that are less vital. So as we already mentioned, our digestive system, right? Salivary glands, stomach, right? This is one of the reasons why you're not supposed to swim till like a half an hour after you've eaten, right? You've just had this big, huge meal. You've got this big, huge cheeseburger that you had for breakfast, and then you go vigorously exercising. If you're vigorously exercising, like swimming a mile out from shore, you are decreasing blood flow to your digestive system, and that food is not going to be not be digested as completely. And if it's not digested as completely, that partially digested food can actually cause intestinal cramping or discomfort. And if you're a mile away from the shore and you're having intestinal cramping, right, that's not the ideal situation for it to be in, right? The skin. If you remember, one of the things we talked about, the skin is a blood reservoir. When you get stressed or someone gets scared, one of the ways you can tell is they get very pale in color because those blood vessels in the skin are constricting to move the blood away from the skin, get it to the heart, to the muscles, to the lungs, and things along those lines as well. All right, I think that is a pretty good and pretty, hopefully, obvious list. Questions on that? All right, let's see what the fancy slide says. So pupils dilate, heart rate, forced contraction, blood pressure, airways dilate, blood vessels to non-essential organs constrict, blood vessels to heart, lungs, skeletal muscle, liver, and adipose dilate. Releasing the glucose, we talked about that from the liver and the adipocytes, increasing blood glucose, decreasing digestive activity. Excellent. All right, questions on that? All right, so let's talk about the parasympathetic. Now, again, remember one of the things we talked about is it's antagonistic to the sympathetic. What that means is that, excuse me, what that means is that anything that the sympathetic does, the parasympathetic does the opposite. So if the sympathetic increases heart rate, parasympathetic decreases heart rate. So one of the easy ways to remember what the parasympathetic does is it just does the opposite of what the sympathetic is. And again, remember we use the rest and digest as our mnemonic for that, but we said there are some better ones. Some of the mnemonics that are used to remember the parasympathetic, remember we talked about being housekeeping types of activities. One of the more accurate mnemonics for it is SLUD. SLUD stands for salivation, lacrimation, urination, digestion, and defecation. Salivation, of course, is producing saliva. Urination, defecation, voiding of urine and feces. Digestion. What's lacrimation again? Crying, producing tears, absolutely. All of these are processes, big major physiological processes that are controlled by the sympathetic nervous system. And notice also, one of the big key differences between our parasympathetic and our sympathetic is, remember, as we talked about, our sympathetic is big global effects where they all happen together. They're all working together. Whereas notice effects. Our parasympathetic are local, discrete effects. Right? When you're sitting there at Ruth Chris and they bring out that amazing looking steak, you start to salivate as a result of that. But do you wet yourself? Do you cry as a result of them bringing that steak out to you? No. Now, you may cry when they bring you the bill a little bit later on, right? But these are processes that are discrete, they're local. It's possible to produce saliva without producing tears, right? Another big important, there are three big decreases. I don't remember if I've told this story in this class or not, but I have a former student. She took me for both 430 and 431. She went through the nursing program at American River College, is currently a nurse now. Great former student of mine. I got to actually go to her pinning ceremony. It was a lot of fun. And her boyfriend have a cabin up in Tahoe. That they like to go up there, and they would study during the weekend. She'd be studying, and he would roam the countrysides up there in Tahoe. And one of the things that he loved to do was pick and eat wild mushrooms. And in the 3 years that actually really in the 1 year of having her in my class, not once, not twice, but on 3 separate occasions, the paramedics had to be called up because he had eaten the wrong type of mushrooms and was basically eating a poisonous mushroom and had to be rushed to the emergency room because of it. What those poisonous mushrooms do, and again, they must be delicious since he keeps continuing to do that, is they stimulate the parasympathetic nervous system. So notice there are 3 big decreases. So you eat that wild mushroom and your heart rate goes down. Your airways constrict. Your pupils constrict. And not only are your airways constricting, but you're producing more saliva. Tears are running down your face, right? So you have all of these horrible effects, right, that don't feel necessarily very good. And yet, like I said, 3 times in the year she was in my class did he do this. So they must be incredibly delicious mushrooms. So there you go. We have those 3 big decreases. Again, remember, we also mentioned how the parasympathetic is the engorgement of erectile tissue. So another one of the mnemonics they sometimes use is they use feed, breed, and grieve, which almost rhymes. Feeding, obviously, all of the digestive functions, breed for the arousal, and grieve for the tears. Some taste like chicken when cooked. Well, then just eat chicken. Chicken's not going to kill you the same way a poisonous mushroom is. All right, excellent. And again, less exciting, exactly. So your book's got a nice table that talks about the physiological responses. And like I said, I think the physiological are pretty simple and pretty straightforward. So hopefully they are fairly obvious. All right. Questions on that? All right, so we have done the easy part. We have done the physiology. What that leaves us with is the anatomy. Now notice here, we have that basic pathway that we have already learned and talked about for the autonomic nervous system. 2 neurons to get from central nervous system to our effector. A preganglionic neuron starts in the central nervous system. Let's go ahead and write this all down because, again, repetition is important. So it is a 2 neuron pathway. Neuron 1, preganglionic. There we go. Excellent. And let's write out everything we know about it. Its cell body is located in the lateral gray horn of the central nervous system. Of the spinal cord, let's be specific, which is in the central nervous system. Excellent. And again, we said most. So we should throw that back in there as well. Axon synapses at the autonomic ganglion. Its axon is myelinated. Its axon, or its synaptic end bulb, releases acetylcholine. And it is always excitatory. Excellent. So that's what we know about all preganglionic neurons. We also know they're postganglionic neurons. Their cell bodies, let's be specific, I want to keep is, actually I guess I gotta say neuron then, since, there we go, is located in the autonomic ganglion. Its axon synapses at the effectors, which we know are going to be smooth muscle, cardiac muscle, and glands. Its axon is unmyelinated. Its synaptic, well, some let's say some neurons, synaptic end bulbs, release acetylcholine, and some release norepinephrine, and some synapses are excitatory, and some are inhibitory. Excellent. So, no new information here. This is the stuff that we have already talked about and we know is going to be true for both our sympathetic and our parasympathetic pathways. So again, we are clear that it is a true neuron pathway, and that is true for both sympathetic and parasympathetic. However, there are some differences between these two as well. One difference is that where in the cell bodies are located, which ganglion, where in the central nervous system they start. Also, how many synapses. I say branching here, but really this also, by branching, we also can mean how many synapses the neuron forms. If you think about it, as we mentioned, our sympathetic nervous system, as we talked about, has big global effects. To do that, one of the ways it can do that is by forming many synapses. So one neuron talks to 100 of other neurons, those hundreds talk to thousands more, and we get big global effects. Whereas with our parasympathetic, nervous system, remember we said we have local effects, discrete. Discrete local effects, and so forms few synapses. So when I say branching, I'm really also talking about how many synapses these are going to form. And they're also going to be different in which neurotransmitter they release. There you go. All righty. Questions on this? All right, then let's look at our two pathways. And let's actually do it this way first. Let's go back to our whiteboard. Everybody remembers what this looks like. And I think I've taken a couple pictures of this, but I'll take one last one just to make sure. So we can delete it. And we can do something similar to what we did before. A line, let's make it black. Chop everything in half. And up here, we will start with our, oops, no, I don't want that to be orange. Our sympathetic pathway. All right, now, of course, we know we need to start in the central nervous system. So there is my spinal cord. We know we need to start in the lateral gray horn. Got my spinal cords just keep getting worse and worse, and I would like to apologize for that. Excellent. And let's go ahead and draw our roots just for the heck of it. There's a dorsal root with the dorsal root ganglion and the ventral root, which we do care about. All right. We know we are going to start with a preganglionic neuron. There are some things we already know about it. Its cell body is located, actually, let's do this way. Cell body is located in the central nervous system, specifically the lateral gray horn, the spinal cord. And we know its axon is myelinated. Those are things that we definitely know about. And again, those are true for both of these. However, there are two big important differences between our preganglionic neuron and the sympathetic versus the parasympathetic. One thing that we talked about, it is going to branch elaborately. Or again, form many synapses. To give us that big global effect. But here's the other big difference between the sympathetic and the parasympathetic. In the sympathetic nervous system, the ganglia, or let's be more specific, the sympathetic are close to the central nervous system. So that sympathetic ganglia, and let's go ahead and use purple for this one, and then give me a purple circle, is going to be close to the central nervous system. If it is, actually, let's do it this way. Let's spread it out a little bit. Perfect. If it is going to be close to the central nervous system, then that means that the length of the preganglionic axon is relatively short. It doesn't have to travel as far to get to its destination. So we have a, oh, I guess that's the other thing we know that's true for both of them, all of them, multipolar. No, great question, Ryan. It doesn't mean that the effect is more immediate. I think the advantage of having it close, again, has to go from the spread. So by having the ganglia close by, it's then maybe easier to get a more diverse spread from to where you're going to go to many different destinations. So we have a multipolar neuron whose cell body is located in the lateral gray horn. Let's give it its dendrites so we know it's multipolar. Its axon comes out the ventral root to the spinal nerve, and then again is going to branch dramatically, forming many synapses. We'll just focus on one of those synapses. And of course, we know another way that these two systems are alike is that it always releases acetylcholine, and it is always excitatory. All right. Where it then communicates with our postganglionic neuron. So again, I'm just showing one of the synapses here, but again, remember, it's going to form many synapses. Our postganglionic, oh, hold on, let me sneak this here and do that. So it gives me a little more room. Perfect. So over here, we can write things we know about our postganglionic neuron. Its cell body is located in the ganglion. Axon is unmyelinated. It is also multipolar. So those are, again, things that we know are true. And we also know it's going to synapse at the effector. But let's talk about the parts that are different about it. Again, it is a sympathetic neuron, so it is going to branch elaborately, or again, form many synapses. And because the sympathetic ganglion are closer to the central nervous system, the length of the axons is relatively long. So it has to travel a further distance to get to its effector, where it's going to form its synapse. And of course, as we know, effectors are smooth muscle, cardiac muscle, glands. Here in our sympathetic pathway, most of our postganglionic neurons are going to release norepinephrine. Release norepinephrine. Of course, most is not all. So there are a few that release acetylcholine. And of course, as we know, oh, so hold on, whoops, shucks. Some inhibitory and some excitatory. So those are things that are true for all of them, but this we need to rewrite. So let's cheat. Erase that.
Quick question.
Yes.
For the post, I thought one of them is global, one of them is local. So where you said that the branch forms many synapses, is that still considered local?
No, remember, parasympathetic is local, whereas sympathetic is global. Our sympathetic pathway is global. So in our so in our sympathetic pathway, both the pre and the post ganglionic branch elaborately.
Gotcha. Okay.
The most release norepinephrine, but a few release acetylcholine. But yes, I just hadn't gotten to that drawing yet, but you are right. Yes, again, we are going to get this elaborate branching where it's going to form many synapses with many effectors. Big global branching for big global effects. Now, again, I appreciate my drawing skills are not the greatest here. So let's look at the pretty picture from your textbook. Here, notice is our sympathetic pathway. Whoa, didn't mean for that to happen. Our sympathetic pathway. So again, notice our preganglionic neuron always releases acetylcholine. This is always excitatory. We know that is always true. But where this is different is that most, oops, no, hold on. Most release norepinephrine, where only a few are going to release acetylcholine. And again, notice the ganglion is close to the central nervous system, so our preganglionic neurons are typically short, whereas our postganglionic axons are typically long. And both branch elaborately. Because again, our goal here is big global effects. All right. Now, let's compare that to our post-ganglionic pathway. I mean, not our post, our parasympathetic pathway. And again, where we want local effects more discrete. Again, there are some things that are going to be the same. We still are going to mostly start, oh, needs to be black. So we're still starting in the spinal cord. Put that there. And so let's draw the rest of it. And again, notice I've just been drawing all of this to the right of the spinal cord. Obviously, the exact same things happening on the left side as well. So there's my ventral root, my dorsal root, and my dorsal root ganglion. So we have that, excellent there. And they come together and they form our spinal nerve. Perfect. Excellent. All right. And again, redundancy because it's important. Our preganglionic neuron. Oops. There we go. cell body in the central nervous system. And again, specifically in the lateral gray horn of the spinal cord. So that's not changing. And the axon is myelinated. And it's multipolar. All of that is the same. But one of the big differences that we see here is in the parasympathetic. Our parasympathetic ganglia are closer to the effectors. In fact, with our parasympathetic nervous system, and actually I'm going to have to cheat a little bit to give ourselves a little bit more room. So I won't exaggerate it quite as much. But in fact, some of our parasympathetic ganglia are so close to the effectors, they're actually in the walls of the effectors. Some are actually in the walls of the effector. For instance, some of the ganglia, the parasympathetic ganglia for the heart, are actually located within the wall of the heart. So they're either right next to or actually inside the effectors. So if they're right next door or actually inside the effectors, then not surprisingly, when we look at these neurons, they are going to the relative length of their, oops, I don't want that to be that big. Relative length of the preganglionic axon is long. And remember, here we want our discrete innervation, so it does not branch. Discrete, let's say it that way. Branching, and it forms few synapses, right, to give us a local effect. So if we were to draw this, so again, we're still out here in the lateral gray horn. We still have our multipolar neuron. Its axon still comes out the ventral root to the spinal nerve, but it travels a much longer distance before it synapses. And it is much more local and discrete in its synapse formation as well. Now, again, this is still an autonomic pathway. So again, it is still always releasing acetylcholine, and it is always excitatory. Our preganglionic neurons are always the same that way. But our postganglionic neurons, and again, let's write what we know about those. Postganglionic neurons, cell body located in the ganglion. Axon is unmyelinated. And it is still multipolar. It is still a motor neuron. All of those things are true. However, again, the key difference is again, because these ganglia are right at or actually inside the organ that they're affecting, the relative length of the axons are short. And again, discrete branching. They form few synapses. And that's what gives us our discrete local effect.
Can you move that part up because I can't see anything for some reason?
Yeah, hold on. Let me draw the neuron and then we'll do that. So there's our preganglionic neuron, postganglionic, I'm sorry, with its dendrites and its axon, going a short distance. Can you not see any of it or just not see the bottom of it?
I can't see any of it.
All right, how about now?
Yeah.
Okay. All righty. One last way these are different. All parasympathetic postganglionic neurons release acetylcholine. Now, it is still true that some are inhibitory and some are excitatory. But notice in our parasympathetic pathway, all neurons, both pre and post ganglionic neurons, all of them release acetylcholine. The distance from the cell body to the synapse does not necessarily determine how branched it is. That is correct. But again, if you think of it, because a lot of times if you think of these pathways as driving directions, right? If you go out to your main street, say you happen to live right off of Douglas. If you go to Douglas, and that's where from there you branch, you know, in Roseville, you have lots of different locations where you can go from there. So I think again, if you think of it from an engineering standpoint, the advantage of having the ganglion close by is it is going to allow you to be able to branch out further from there and have more local effects. If the ganglion that you synapse at is right next to or even inside the heart, then chances are you're not going to be able to go very many places other than the heart. So I think that's probably, again, why things are is always one of those questions that's hard for us to answer. But I think if you think of it from an engineering standpoint, yes, the closer the ganglion is to the effector, the more local the effect is going to be. All right, questions on this? This doesn't make sense. You need to let me know now because it is about to get a whole lot worse.
Yeah, quick question.
Yes.
So the sympathetic and the parasympathetic, they both have the pre and the post neurons, correct?
Yep, and that's one of the things I tried to emphasize here on this illustration. The things I wrote in black are the things that are true for both the sympathetic and the parasympathetic nervous systems. So both have two neurons. Both preganglionic neurons always release acetylcholine, are always excitatory. Both preganglionic neurons are myelinated, where the postganglionic neurons are unmyelinated. So absolutely, there are a lot of things that definitely are similar. And then the things that I color-coded are the things I tried to emphasize that were different. So I did try to emphasize that with the coloring that we used on this particular illustration. And I'll post this one and the previous one that I did as well. All right. But again, you don't have to just rely on my pretty pictures. We have the pretty pictures from your textbook that show this as well. Notice the difference here between our sympathetic and our parasympathetic. Very discrete, very limited branching. Notice our autonomic ganglion is way closer to the effector, whereas before it was closer to the central nervous system. So we have a much longer preganglionic neuron, a much shorter postganglionic neuron. And notice here in our parasympathetic pathway, all of the axons release acetylcholine. All preganglionic, all postganglionic release acetylcholine. All right. And again, notice you've got that nice table in your textbook where if we come back and look at this again, remember when we looked at it the first time, we were emphasizing the difference between the the somatic and the autonomic. But notice now we can see the difference between the sympathetic and the parasympathetic. Parasympathetic ganglia are closer to the effectors. Postganglionic neuron always releases acetylcholine. here in our sympathetic pathway, most postganglionic neurons release norepinephrine. Again, key word on this one is indeed most. Right? Most release, whereas here, absolutely positively, all release acetylcholine. And notice also, one of the ways, because again, our sympathetic is about global effects, whereas our parasympathetic is about local effects. One of the ways we see how that is emphasized is, as we also talked about, our sympathetic is going to cause our adrenal gland to release that epinephrine and norepinephrine into the blood, giving us that longer lasting global big stress response. Whereas, as we know, the adrenal gland is not innervated by the parasympathetic pathway. So I've looked at this a couple of different ways. Questions on this? Because like I said, if this isn't making sense, I'm dead serious. It's about to get a lot worse. Yeah, definitely, absolutely. Your spinal cord is part of your central nervous system. Damage to the spinal cord affects all things, right? Touch, motor, and autonomic control of things, absolutely. So now that paraplegic, not only can they not feel their legs, not only can they not move their legs, but the hairs on their legs can't stand up. They can't dilate or constrict the blood vessels, right? Problems along those lines. Arousal can be an issue. Defecation and urination can be an issue. Yep. Spinal cord is important, which is one of the reasons why it's so protected. All right. We have, how are we on time? Oh my. All right. Yep, absolutely. Housekeeping would be as well. Although, again, remember, if you think about legs, there aren't that many housekeeping effects in the legs. Housekeeping tends to be more, internal organ type things, right? If you think about the muscles, if you think about the skin, right, the blood vessels that are in the legs or in the arms, those are pretty much just controlled by the sympathetic. So we got motor control of the muscles, somatic motor control of the muscles, but pretty much everything else out on the arm is sympathetic. Parasympathetic is more internal, centralized. All right. Here is where things get fun. This scary looking monster. Now, when you first looked at this, which hopefully was this weekend, hopefully you're not seeing this for the first time now, but when you do look at this for the first time, it can be a bit intimidating at first. These scary octopi type looking illustrations. But surprisingly, these illustrations actually do a pretty good job of giving us a basic schematic of our autonomic nervous system. Notice a couple things that should jump out to you right off the start. Notice, for instance, if you are going to control the smooth muscle of your eyes, there are two neurons in those pathways. In the parasympathetic pathway, we have a longer preganglionic neuron because we know the ganglion is much closer to the eye, and a shorter postganglionic neuron. Whereas if we're getting to the eye in the sympathetic, the ganglion are very close. So we have our dark preganglionic neuron, which notice branches elaborately, but if we're getting to the eye, we'll come up to here, synapse. So a relatively short axon, and then a longer axon to get us to the eye. Notice the skin, the adrenal gland. Those are things that are not found on this side because we know things like the skin, things like the adrenal gland only receive input, from the sympathetic nervous system, and the parasympathetic nervous system does not innervate them. So while this can be a bit intimidating at first when you look at it, we can see that it actually has, right, ganglion right next to the organs, ganglia inside the organs, long preganglionic neurons, short postganglionic neurons, short preganglionic neurons, longer postganglionic neurons. We can see some of that basic anatomy that we've been talking about in these illustrations. And here we see a little bit more of an elaborate one. Yes, the lighter colors are the postganglionic neurons, the darker colors are the pre. But like I said, this is a pretty basic schematic. So when you look over here at the parasympathetic and what it looks like on this illustration, and then you look at what the parasympathetic looks like on this illustration, you can see that, like everything we talk about in the class, it's more complicated than just this basic schematic. And even this is, not nearly as complicated as it really is. Oh, nope, adrenal gland. Well, we'll talk about that pathway. That is a trickier one, yes. But you get the idea where there are indeed two neurons to these structures. All right? So here is the parasympathetic, and notice parasympathetic doesn't go to the adrenal gland. It doesn't go to the skin, things along those lines. And so this is what we're looking at. Now, obviously, there are going to then, as you can see, be some big differences between the sympathetic and the parasympathetic. Pathways. When we talk about what those are, one of the big differences, as you can see, is where they start in the central nervous system. Notice they don't overlap inside the central nervous system. They are looking at they are located in different locations. Therefore, the structures that are formed by the axons are different. So we're going to need to identify those different structures, those different pathways, right? So we can think of this as the pathways involved in this. To get to their destinations. So we're going to have different structures, different pathways, and of course, different ganglia for the sympathetic and the parasympathetic. So what we need to do then is focus on these differences and describe the pathways. And that's what we're going to do next. We are going to start first with, and I'll go ahead and leave this here. So let's bring this down so it's out of the way. And what we're going to do is talk about the parasympathetic first. Notice from an anatomical standpoint, we call the parasympathetic nervous system the craniosacral branch of the autonomic nervous system, because if you notice, it starts in the cranial and sacral regions of the central nervous system. So notice all of our preganglionic neurons are either located up here, and let's highlight it with a highlighter, are either located up here within the brain stem, coming off of cranial nerves, so that's the cranial part, or down here in the lateral gray horn of the sacral region of the spinal cord. So since they have two different locations of the central nervous system they come out of, anatomically we refer to it as the craniosacral branch. And so what we're going to need to do is identify these different regions of the central nervous system, identify the structures that are made by the axons, identify the ganglions, and identify which effectors they're going to affect and influence. Now, like I said, the good news is, again, our parasympathetic is more discrete. So it is a simpler pathway. Or simpler anyway. And really, there are 6 main pathways we will identify. And so that's what we're going to do when we come back after our last break. We're going to come back after our last break, and we're going to identify the 6 main pathways that we will find in the parasympathetic nervous system. And easily these could be essay questions that you could potentially have or need to describe on the exam. All right? Questions on that? All right. Things are about to get fun, so this is a good stopping point. Let's go ahead and take our next break. I'll leave all these words here on the screen so we can let that sink in. Looks like it's 10:30, so let us restart at 10:45, and I will start the recording at that point. All right. Any other questions before we take our next break? All right. See you in 15 minutes. All righty. So let's have some fun. Let us talk about the anatomy of this and draw it out, not draw it out, but write it out and make some sense of it. And then we'll come back and look at this picture and make sure that it makes sense. So for this, we need to go back to our whiteboard, make sure that I save this one, and then clear it. All right, so any questions before we get started? All right, so what we need is some columns to make sure that we understand what our pathways are going to be. So what we are going to start first is with our preganglionic neuron. With our preganglionic neuron. And again, we should probably emphasize that we are looking at the parasympathetic nervous system with this. For our preganglionic neuron, there are basically 2 pieces of information we need to know. We need to know where the cell body is located. So let's shorten that a little bit. And we need to know the structure or structures formed by the axon. Okay. For our for our postganglionic axon, neuron, we need to know the ganglion, where it is housed. So we're actually, let's say it this way. Let's be specific, where the cell body is located. So I'm going to need to put that down there for now. Let's put this down there for now as well. Where the cell body is located. Remember, one of the things we talked about with our postganglionic neuron is they are either in or right at the effector. So when it comes to their axons don't really form anything. All of their axons basically just form postganglionic or parasympathetic nerves is all they're going to form. So really for all of them, all form parasympathetic nerves. So that isn't really anything that we need to worry about that is going to differentiate them. But what we do care about is what effector it is going to synapse at, and then probably also want to think in terms of the effect that it is going to have as well. All right. Now, let's start with the easy part of this. With the parasympathetic nervous system, remember, from an anatomical standpoint, we refer to it as craniosacral. And the reason for that is there are only 2 locations in the central nervous system where the cell bodies are going to be located. They're either going to be found in the brainstem, or they are going to be found, and I'll sneak that over here. We can write it down here. We may end up moving it, but for right now, I'll put it down low here. In the lateral gray horn of the sacral spinal cord. So that's kind of where we left off last time. All right. Now, if we go back and look at that picture, and we'll do that in just a second, for our brainstem, what we will see is, in particular, there are 4 cranial nerves that contain parasympathetic axons. Oops. Nerves. There we go. That contain parasympathetic axons. Anyone cheat and peek and look back at our picture and tell me which cranial nerves those are? Excellent. One of them is indeed the vagus, cranial nerve 10. Vagus. Oops. What other ones? Hopefully some of you have printed out the notes, have them in front of you, can figure it out. Take a peek. I'll wait. 3, excellent. What was 3 called again? Ocular motor, excellent. Excellent. What other one? Facial, what number was that? Facial 11? 7, there you go. 7, oops, let's stick with the Roman numerals. Facial, and the last one was 9, excellent. Glossopharyngeal. Oops, hold on. So fair, God, I'm way off on that. There we go, glossopharyngeal, excellent. So there are 4 cranial nerves that are going to be formed, that are going to contain this, and actually that should probably go over here into our structures formed by the axons. Cranial nerve 3, cranial nerve 7, cranial nerve 9, and cranial nerve 10. But of course, if their axons are forming these, then that means that the cell bodies are located in ganglia. Pardon me, not ganglia, in nuclei in the brainstem of the brainstem, say it that way. And of course, for nuclei 3, or nucleus, let's say it that way. 3, nucleus for 7, nucleus of 9, and nucleus of 10. So in our brainstem, we have these nuclei where the cell body is going to be located, and then their axons are going to form cranial nerves 3, cranial nerves 7, cranial nerves 9, and cranial nerves 10. So cell bodies are located in the brainstem, in the cranial region. Excellent. All right. Now, when it comes to the postganglionic, when it comes to the parasympathetic ganglia, All these cell bodies are located in parasympathetic most of them, and there's that pesky word most, as we talked about, are right at or in the organ, the effector. Now, if we've learned anything, it's that anatomists love to name everything. But when it comes to a tiny cluster of nerve cells inside the wall of our heart, even our anatomists balk at naming that. So most of the ganglia are just simply referred to by a basic term, and that basic term is a terminal ganglion. In fact, there are only 4 parasympathetic ganglia that have names. There are hundreds of parasympathetic ganglion, but there are only 4 that have names. And those 4 that have names are ciliary. The next one is pterygopalatine. The next is submandibular. And the next is otic. So there are only 4 ganglia that have names in the parasympathetic nervous system. The rest just use a basic term that we call the terminal ganglia. And these are the only 4 that have names. Now, if these 4 have names, that means that they probably innervate something pretty important. And that is the case. Our ciliary ganglion innervates the smooth muscle of the eye. So if you think about it, remember our ocular motor controls motor, as we talked about, of the eye. So this preganglionic neuron that starts in the nucleus of 3 and forms cranial nerve 3, the ocular motor, goes to the ciliary where it synapses, and then the ciliary innervates the smooth muscle of the eye. So if you want to dilate or constrict your eye, if you want to change the focus of your eye, this is the path. You start in a nucleus associated with cranial nerve 3, out cranial nerve 3, to the ciliary ganglia, to the eyeball. Pterygopalatine is, again, one of those alphabet soup terms that you are going to need to know. But again, the name tells you a lot about it. Palatine refers to what? Anyone know what palatine refers to? What does that sound like? Palatine bone, exactly. So palatine bone, the hard palate, has to do with the nasal cavity. All right, roof of the mouth. And in fact, pterygo. When I say pterygo, what does pterygo sound like? When I say pterygo, what do you hear in your head? What does it remind you of? Maybe pterodactyls? That's what it reminds me of. And what's special about pterodactyls? Well, let me rephrase that. What's the second thing that's special about pterodactyls? Because when I say what's special about pterodactyls, the first thing everyone says is that they can fly. Absolutely. They can fly. That is one of the special things about them. But what's the second special thing about a pterodactyl? How can you easily identify a pterodactyl? Aside from the wings. What's their other big key characteristic that they have? Right? If you're not sure, go check your dinosaur chicken nuggets. There's a pterodactyl in there. And what's special about it? huge noses, huge beaks. And that's what pterygo means. Pterygo means beak. So this is that nasal cavity. This is the roof of the mouth. And the other thing it innervates is your tear ducts. So that our pterygopalatine is the ganglion that causes us to cry, produces mucus in our nasal cavity, right? Maintains the nasal cavity, the roof of our mouth, and our tears. It is innervated by axons of the facial nerve. But if you look closely at our picture, you will notice that not all the axons of the facial nerve go to the pterygopalatine ganglion. Some of the axons of the facial nerve also go to the submandibular ganglion. And the submandibular ganglion innervates salivary glands. Now, as it turns out, you have 3 pairs of salivary glands. You have your parotid, submandibular, and sublingual. And again, you don't need to know those for this exam. You'll learn that when we get to the digestive system. But with 3 pairs of salivary glands, it turns out you actually have 2 ganglion that innervate those 3 pairs of glands. So the submandibular innervates salivary glands, and the otic also innervates salivary glands. And the otic is innervated by the glossopharyngeal nerve, cranial nerve 9. So notice, all of 4 of our named ganglia all involve our head. So if you want to focus your eyes, again, that doesn't increase saliva production. You can increase saliva production without crying. We have very local discrete effects because we have these local discrete pathways, just basically one way to get to all of these destinations. All right. So those are the ones that have names. The rest of our ganglia, remember, do not have names. The basic term for them are just terminal ganglia, including all of the output of our vagus nerves. Our vagus nerve, remember, as we talked about, our vagus nerve is responsible for 90% of our parasympathetic output. And all of the output coming out of our vagus nerve all goes through terminal ganglia that are right at or in the organ affected. So even our sad, lonely anatomists haven't bothered to name any of them. And as we talked about, our vagus nerve innervates most ventral organs, say it that way. That's what we said last time when we were talking about it in the cranial nerve standpoint, but now we'll be more specific. Heart, lungs, stomach, spleen, pancreas, liver, small intestine, and the proximal large intestine. So if you want to control any of those organs, heart, lungs, stomach, spleen, pancreas, liver, gallbladder, small intestine, and the proximal part of the large intestine, it is the vagus nerve that controls it. So notice, if you cut that vagus nerve, you're in trouble. because it controls a whole lot of stuff. But notice again, there's that key word most. There are some organs that weren't on that list. Really, three main groups. The first are the reproductive, organs, right? Penis, vagina, ovaries, testes, depending on your flavor. Urinary system, bladder, ureters, urethra, kidneys. And notice the distal large intestine. And by that, primarily, we mean the rectum and the anus. These structures are also controlled by the sympathetic nervous system, but not by any of your cranial nerves. Now, remember, as we mentioned, there is the craniosacral branch of the spinal cord, and we'll cheat and do a light highlighter to kind of differentiate these, because this up here is the cranial part, and then this down here is the sacral part. And so that sacral part is going to come out of the lateral gray horn of the spinal cord. As we know, it is first going to form the ventral root, and then it is going to form the spinal nerve. But then from the spinal nerve, it branches to form a structure that we call the pelvic splanchnic nerves. Now, I will tell you that some textbooks use the term just pelvic nerves, and I will accept that. Pelvic nerves is acceptable. I will explain to you why I use the term pelvic splanchnic, because that's what A was originally called, and there's a reason for that. So we'll do that as well. These pelvic splanchnic nerves then go almost all the way to the organs, where they're then going to terminate on terminal ganglia. And then, of course, those terminal ganglia innervate the reproductive organ, urinary organs, and the rectum and anus. Yeah, exactly. Good old Bob Splanchnick was the first person who described it. Actually, I don't know if Splanchnick is a last name, but no, the reason is when we get to the sympathetic nervous system, there are some Splanchnick nerves there. So what helps me to remember is Splanchnick with an S, sympathetic, pelvic splanchnic, PS, parasympathetic. So that's how I remember the two apart. So both of those are going to be structures we're responsible for identifying on the exam. And that mnemonic of remembering the names helps me to remember which branches they're associated with. I've done it here with what hopefully is an intuitive schematic. But if we go back to the picture from the textbook, we can see exactly what we just described. Notice route 1 in the parasympathetic nervous system starts in a nucleus in the brainstem, comes out cranial nerve 3, goes to the ciliary ganglia where it synapses, and the postganglionic neuron goes to the eye to control the smooth muscle of the eye. Route 2, starting with the nucleus in the brainstem, coming out cranial nerve 7, going to the pterygopalatine ganglia, which innervates where it synapses, and then the pterygopalatine synapses postganglionic axon innervates the lacrimal gland or the nasal cavity. Pathway 3, we start in a nucleus found in the brainstem, come out cranial nerve 7, After cranial nerve 7, this time to the submandibular gland, where it innervates our salivary glands. Pathway 4, nucleus found in the brainstem, out cranial nerve 9, glossopharyngeal, synapsing on the otic ganglion, where the postganglionic axon goes to a salivary gland. Pathway 5, starting with the nucleus in our brainstem, coming out the vagus nerve. From the vagus nerve, we can go to the heart, we can go to the lungs, the liver, the gallbladder, the stomach, the pancreas, the spleen, the small intestine, and the proximal part of the large intestine. Notice their ganglia are right at, or actually inside the organ. So they don't have names, they just synapse on a terminal ganglion, and then that terminal ganglion innervates that specific organ. But notice our 6th pathway doesn't start in the brainstem. It starts in the lateral gray horn of the sacral spinal cord. So that's where the cell bodies are located. Their axons come out the ventral root, form the spinal nerve, and then form the pelvic nerves, or pelvic splanchnic nerves. And they go to the rectum and anus. They go to the urinary system. They go to the reproductive system. So 6 discrete pathways providing all of our autonomic control, local discrete control of these specific organs by these specific paths. And again, we've done it a couple times. Let's do it a third. Only 4 named parasympathetic ganglia, and they're associated with our 4 cranial nerves. Of course, it's a little bit of a cheat because 10 doesn't have any named ganglia and 7 has 2. But we do have 4 cranial nerves. We do have 4 nuclei. And again, all those named ganglia are in the head. Ciliary goes to the eye. Here, this one's a little nicer. Pterygopalatine goes to the nasal cavity, goes to the lacrimal gland. Submandibular goes to salivary glands. Otic goes to salivary glands. And notice here we see more clearly submandibular goes to the sublingual and submandibular otic goes to the parotid. But as I mentioned, I'm not making you know the 3 names of the 3 different types of salivary glands. So as long as you know both submandibular and otic innervate salivary glands, you are fine.
Question?
Yes.
Does the number 7, is that considered as 2?
Well, see, what you got to remember, cranial nerve 7 is a nerve made up of many axons. Some of those axons are going to go to the submandibular ganglion. Some of them are going to go to the pterygopalatine ganglion. And some of them, remember, are going to go to the face so that we can control the muscles of the face. Some of those axons go to the tongue because they carry taste sensation in. Right? Remember, it's a bundle of axons. So, you know, a single nerve can have thousands of axons in them, and they're not all going to go to the same location. So different axons are going to branch to go to different locations.
Okay, because I just counted them. You said there was 6, but for some reason I keep seeing only 5. That's why I'm asking.
Well, on this screen, you only see 5. 1, 2, 3, 4, and 5. But remember, we have the sacral at the bottom. That is our 6th.
Oh, okay, got you. Okay, so then 7 is considered as 2, not just 1.
Yes, going to the pterygopalatine is one pathway. Going to the submandibular ganglion is a different pathway.
Got you. That's what I meant. Yeah. Okay.
All right. That 5th pathway is our vagus. And again, it's 5th just as we're counting our way down. But remember, about 90% of our parasympathetic output. And it is our wanderer. Remember, it's our longest cranial nerve. It goes to the heart, lungs, liver, gallbladder, stomach, spleen, pancreas. You can see the spleen in this picture. It's missing the spleen. Large intestine, small intestine, and the proximal part of the large intestine. And again, none of the ganglia, even this goes to the majority of the organs, none of these have names to them. They're just terminal ganglia. Now notice, Technically, the ones that are within a wall of an organ are sometimes referred to as intramural, right? Intramural, if you do intramural sports, it's sports within the campus, within the walls. Intramural means within the walls. I mention it because your book mentions that term. I don't care if you know it or not. Terminal ganglia is a good generic term, and I'm fine with that. But as Kendra just pointed out, we cannot forget about the sacral portion of the craniosacral parasympathetic nervous system. Right? They come out the spinal cord, they form those pelvic splanchnic nerves, and those pelvic splanchnic nerves go to the rectum and anus, to the urinary system, and to the reproductive system. Ready? Questions on that? All right, excellent. Just that easily, then, we have done the parasympathetic pathways. Like I said, the advantage of these are they're simple, they're straightforward, they're discrete. Now, I know if you go back to that chart, it may not seem terribly straightforward, but compared to what we're going to do with the sympathetic, it is very discrete, very straightforward, very basic. All right. Questions on that? Because like I said, it only gets worse from here if this doesn't make sense. All right, so let's go back and look at this one more time. Here are those 6 possible pathways to get to discrete innervation. So if I ask you to describe the structures, describe the pathways of how the parasympathetic innervates the nasal cavity or innervates the smooth muscle of the eye or innervates the heart or something like that, you should be able to answer that question. All right. Excellent. All right. Stunned silence means that we completely understand. Please make the test harder. So excellent. We will move forward. Now we get to look at the green side of this, the more scary looking monster octopus portion of this. Again, here is our very basic schematic. Here is one example of a little bit more of a specific example. And here we see that there are some, again, big anatomical differences. For starters, from an anatomical standpoint, our sympathetic is known as the thoracolumbar. Branch of the autonomic nervous system. Because it only starts within the lateral gray horns of T1 through L2 of the spinal cord. So notice all of, within, wow, the lateral gray horns of T1 through L2. So just thoracic and lumbar, so thoracolumbar branch of the autonomic nervous system. The second thing that should stick out from you from this picture is that there are 2 main types of sympathetic ganglia. There are the chain ganglia, which are also known as the paravertebral ganglia, because they are parallel to the spinal cord. And they are paired. If they're going to be parallel, they're also paired ganglia. I know this picture doesn't show that, but the next one will. They're paired ganglia, meaning you have 2, one on the left and one on the right. The second type are what are called the collateral The picture here makes it look nice because they are over here to the side. However, it's a little bit tricky. These collateral ganglia are single ganglia. They're unpaired. And they're actually located on the midline. Actually in front of the vertebral column. So because of this, they are also sometimes referred to as the prevertebral ganglia. And not surprisingly, and again, the illustration kind of emphasizes this is a little bit, two different ganglia in two different locations with two different functions. Our chain ganglia are going to innervate basically our skin. So if you think about those dermatomes we saw of the body, so the hair, muscle, skin, limbs, and internal organs above the diaphragm. Whereas our collateral ganglia innervate the internal organs below the diaphragm. All right. A lot of words. Let's take a look at the pretty picture and see how this works. Again, this is the thoracolumbar branch. We start in the lateral gray horn of the spinal cord from T1 down to L2. And notice there is no straight discrete pathway. We have a massive elaborate innervation and branching of this. So there are many, many routes that you can take to get to your destination. So it's going to be a little bit messy, but we're going to focus on the primary routes, right? There are plenty of ways that you can get from your house to your work. But that doesn't mean that each day you take a different route. You typically take the most efficient route, but there are dozens of other ones you could take. And the same thing is true here. We will focus on the main routes, but you got to remember there's lots of collateral routes because of the huge, massive innervation. All right. Here, we kind of get a better view of some of those ganglia. Noticing first here, these are the chain ganglia. Again, as we talked about, they are paired ganglia. So we see them lined up together in a chain, connected by a chain. And notice there is a chain going down the other side as well. And because they are to the sides and run parallel to each other, as I mentioned, they are also sometimes referred to as the paravertebral ganglia. As we said here, so there are those chain ganglia or trunk ganglia, sometimes referred to, and they are basically labeled by their specific location. However, it is a little bit more tricky than this. They are paired, so notice here we see a better view where there's some on the left and some on the right. But something interesting does happen. Notice down here at the bottom, we have, and we'll talk about this in just a minute, there are primarily 3 cervical, different numbers of lumbar, so 9 to 12, thoracic, somewhere around 4 to 5, lumbar, somewhere around 4 to 5, sacral, and everybody has 1 coccygeal ganglion. One of the interesting things about the 2 coccygeal ganglia is these 2 coccygeal ganglia, one on the left and the one on the right, fuse together. And when they fuse together, it turns our chain ganglia into this big U-shaped structure. And the fancied word we give this fused structure is the ganglion impar. So where those two coccygeal fuse together, we call them the ganglion impar. Now, as I mentioned, these innervate the head, the limbs, the skin, and all of the internal organs above the diaphragm. As I specifically mentioned, there are 3 cervical ganglia. And here we get a closer look at the 3 cervical chain ganglia. Not surprisingly, they are named based on their location. We have the superior cervical chain ganglion. We have the middle cervical chain ganglion. And we have the inferior cervical chain. And if we look at the illustration here, we can see what these structures innervate. And again, I'm going to write this word primarily because, again, remember, there's lots of branching. Primarily. The superior cervical chain ganglia primarily innervates the head. So notice the lacrimal gland, the smooth muscle of the eye, the mucous membrane of the nose, big global effects. So again, when you're stressed, right, your eyes get dry, your mouth gets dry, your nose dries up, your pupils dilate, all those effects are all happening together. Now notice there is a minor input to heart from this, but again, that's not surprising because there's big elaborate branching. And notice the middle and middle and inferior cervical chain ganglia both innervate the heart and lungs. And in a few individuals, these two will fuse together. So in a few individuals, some rare individuals, they will actually just have 2 cervical ganglia instead of 3. Now again, does that affect the function? No. Right? It's just how the wiring inside the wall is set up. Doesn't really dramatically affect the function at all. It's just an anatomical distinction that has no relevance in function because they were both going the same place anyway. All right. Questions on those? So those are some of the potential pathways that we're going to need to get to some of these different locations. And again, I've written it out, but let's look at it with the pretty words. Right? All innervate skin, smooth muscle and glands of the head, the minor input to the heart. And like I said, middle and inferior innervates the skin and the thoracic cavity, basically the organs above the diaphragm. And sometimes fused together. Well, so then the question becomes, what the heck do the rest of the chain ganglia do? Well, remember, as we talked about, the rest of the chains go to the skin and limbs. So I mentioned somewhere between 9 and 12 thoracic, 4 and 5 lumbar, 4 or 5 sacral, 1 coccygeal. And as you can see, they start in the thoracic, they started in the lateral great horn, of the spinal cord, synapse in one of these inferior chains, and then go to things like the sweat glands, like the erector pili muscle, like the adipose and the blood vessels of the skin and the limbs. And notice, this one is T1 up here, right? This one is L2 down here where the nerves are. But remember, the number of chain ganglia vary. So if I were to highlight this chain ganglia on the exam, do we know with 100% certainty what chain ganglia that is? I mean, that could be thoracic 10. It could also be lumbar 2, right? So there's really no way to know for certain. So we're not going to worry about it. We definitely can tell the first three: cervical, superior cervical, middle cervical, inferior cervical. These three we absolutely positively can identify and name. For the rest of them, we're just going to call them chain ganglia and not worry about identifying each individual one. And conveniently enough, they all do the same thing as well. All the autonomic functions of the skin. So they're going to the arms, they're going to the limbs, they're going to the fat, they're going to the muscle, to the blood vessels of our skin. All right. It's okay if this is starting to get a little confusing. We are going to actually take some time and actually draw these pathways specifically out. And that I do guarantee will be an essay question that we will get to. But I want to go through the basic anatomy first. Here's one last pretty picture that does a nice job of showing us these chain ganglia. Again, and here we see the relationship to them. They're to the side of the vertebrae. So again, that is why they're called the paravertebral vertebrae. And I want you to notice one more thing, layering information that we already know on top of each other. Notice here is that chain ganglia, and notice what we have going to it, those rami communicantes. Remember, the lateral one is the white ramus, the medial one is the gray ramus. The white and gray rami, notice, are coming off of the spinal nerve and going to that chain ganglia. And we'll see why that's important when we're talking about that pathway, that process. And here we see it again. And remember, as I promised you here, we'll see it more accurately. Remember, the lateral one is white, the medial one is the gray, going to the chain. And notice each segment of the spinal cord has a white and a gray ramus and a chain ganglia that it connects to. And the chains are interconnected as well. But we also have three prevertebral ganglia. The superiormost one is known as the celiac. Notice not ciliary, remember that's in the eye. This is celiac ganglion. It is the most superior. The one that's in the middle is the superior mesenteric. And the inferior one is the inferior mesenteric. Now, if the most superior one is the celiac and the middle one is superior, then why the hell is it called the superior mesenteric? Why are these called what they are called? That doesn't seem to make any intuitive sense to me. Anybody know where these three names come from? Close. Part of it could be related to the areas that they innervate, but one of the things that happens is our nerves and our blood vessels tend to travel together. So these large ganglia are located next to major blood vessels. The celiac ganglion is located right next to the celiac trunk or artery. The superior mesenteric ganglion is located right next to the superior mesenteric artery. And the inferior mesenteric ganglion is located right next to the inferior mesenteric artery. So these are all next to major blood vessels, and so they get their names not by their relative location, but by the blood vessels they're sitting next to. Now, as you can see from the illustration, as we've talked about, there is going to be a lot of overlap in their functions. But in general, as you can see, our celiac ganglion innervates the liver, the gallbladder, the stomach, the spleen, and the pancreas. Our superior mesenteric innervates the small intestine and the proximal large intestine. And the inferior mesenteric ganglion innervates the urinary system, the reproductive system, and the distal large intestine. Again, the rectum and the anus. So again, there, as you can see, is overlap because it's a big, massive, overlapping system. But in general, the celiac ganglion innervates the proximal part of the digestive system. So again, liver, gallbladder, stomach, spleen, pancreas. Superior mesenteric ganglion goes to all of the small intestines and the proximal part of the large intestine. And the inferior mesenteric ganglion goes to the distal large intestine, rectum and anus, urinary system, and reproductive system. So there you go. We have identified all of the ganglia, and we have identified the structures that are going to be innervated by those ganglia. So we know what ganglias are and where they go. And that's what this picture shows. Notice we cheated a little bit. All of these over here are going to the skin, and I had to remove the skin on this side so we can't really see it, but we know all of these, and again, all of those are going on the skin as well. We see here that our superior cervical ganglia is going to things in the head. Middle and inferior is going to the heart and the lungs. Celiac ganglion is going to the liver, the stomach, the spleen, the pancreas, right? Our superior mesenteric, small intestine, proximal large intestine, and our inferior mesenteric to the reproductive system, to the urinary system, and to the rectum and anus. So we've identified all the ganglia, and of course, all of these chain ganglia all go to the limbs and the skin. So while it's a big, scary looking octopus, we can see synapses in the ganglion. Red is the preganglionic, X is the postganglionic, going to all of our organs. And so this is what we have to make sense of. While we can see this, there are a lot of structures involved. It's a more complicated pathway than what we saw in the parasympathetic. So we did all that, we did all of that. We talked about that. So this is what I want to finish off on. How are we with time? Perfect. All right. What we are going to do starting in the beginning of class tomorrow is we are going to draw the 4 main sympathetic pathways. And I guarantee that describing one of these 4 pathways is going to be an essay question on your exam. So what I'm going to do is I'm going to give you some destination in the body. Eye, salivary gland, heart, lungs, stomach, spleen, small intestine, large intestine, erector pili muscle, right? Sudoriferous gland. I will give you one of the infinite organs or structures in your body. Okay, not infinite, but hundreds. And you will describe the one of these four pathways that gets us from our sympathetic innervation of that. And that's how I'll ask it. Describe the sympathetic innervation to the X, to the eye, to the heart, to the stomach, to the rectum, whatever it is. Now, your book actually talks about all four. It only emphasizes three of them, so I want to make sure to emphasize four. But how we're going to do that, as you can see from this, is we are both going to draw it and describe it. To help us get a head start on tomorrow, what I want to do is give you the basic drawing of this first. What I want you to do is make 4 copies of this drawing in your own way, however you want to do this. But if you do these drawings first, it will save you some time when we are doing this together as a group. All right, so let's do this. Come back to here, make sure we've saved that, and then I can clear it. Now, obviously, the first place we need to start, no, black, is in the spinal cord. Now, I'm going to cheat a little bit and set this off to the side. We know, and again, I'm going to cheat a little bit more, that there is the dorsal root with the dorsal ganglion and all of those things. But we also know we're talking about a motor pathway, so we don't really care about the dorsal side of this. So when it comes to the drawing of this, and actually I'm going to cheat and move that. I need a little bit more room because I'm a horrible artist. So move this to there, move that to there, move that to there, and move that to there. Excellent. Now, we know coming off the bottom of this is going to be a ventral root. Coming up, we know, and I'll draw it on this side. Dorsal root ganglia comes together and forms the spinal nerve. But we're drawing motor pathway. All we really care about is that ventral root. So we'll focus on the ventral root coming up to our spinal nerve. We know our spinal nerve is going to branch. I'm going to draw these things first, then we'll go back and label all of them. And it branches not 1, not 2, not 3, but 4 distinct branches that come off of that spinal nerve. So let's start adding some labels. This is, of course, our spinal cord. This is our ventral root. This is our spinal nerve. And 4 branches are dorsal ramus, ventral ramus, gray ramus, and white ramus. Now, we know now that the gray and white ramus is going to connect to, oh, let's cheat. There we go. It's going to connect to one of our chain ganglia. And these chain ganglia, so now I'm going to really have to cheat now, we know connect to chains. So we know coming off of this is going to be a chain coming down. Whoops, no. I need that. So a chain coming down to a second chain that's below it, and a chain coming up to a second chain ganglia that is above that. And again, my artistic skills are not the best, but you're just going to have to deal with it. Draw this however you want. If you're better drawing three-dimensional structures, then yay, more power for you. I am not. So, but we can cheat and draw that, and cheat and draw that. Excellent. So those are, so again, this is a chain above. So this chain ganglion above, and again, we know above is ascending. So this is an ascending chain ganglion. And we know this is a descending chain ganglion. That we have down there, coming off of the bottom of that. But we also know that chain ganglia are not the only type of ganglion. We also know that here in front of the spinal cord are our collateral ganglia that are unpaired. Of course, we know there must be axons that connect the two, so we can cheat and draw that as well. Although we won't worry about naming that just yet. But we know there's going to be a connection here like that. And I'll go ahead and warn you right now, there is one more pathway out of this chain ganglion. Oops. That we haven't talked about yet, but we will identify tomorrow. So I want you to put it on your picture as you're drawing it. And oops, there. Excellent. We'll worry about the areas that they're going to innervate later. We're going to put a list over here at the sign, but this is the anatomy. I want you to have some space or something else that you can write on, but I want us to be able to draw these things as we go from here. But this, in its simplest, basic, scariest terms, is the basic anatomy that we need to be able to identify. And so again, well, obviously you're not going to be drawing this on the exam because there's no way for you to draw it. Obviously, if we were doing this in the classroom, I'd probably make you draw it, but we'll describe it in words, and you will describe the pathways that we're going to get, starting from the central nervous system to any possible effector. So you're going to need 4 of these for tomorrow. If you want to have 1 or 2 extra in case you mess up or in case you want to do it again or something like that, then I would encourage you to do that. So make 1 and photocopy it or draw it 6 times so that you know it. Whatever helps you to make sense of this, we will work on this together tomorrow. So this is where we're going to pick up. Tomorrow, we are going to start right off the bat with drawing this picture and working our way through this picture and identifying and drawing those 4 pathways that are going to get to us to every single effector in our sympathetic nervous system. All right. And again, obviously there's clearly more than 4, but 4 basic main pathways with some extra identification. And like I said, I guarantee you one of these questions will be an essay question on the exam. So you absolutely positively need to understand this. Not just because it's going to be on the exam, but like I said, this is how we get to the stomach to control the stomach. This is how we get to the heart when we control the heart. This is how we get to the kidney and everything else. So when we talk about those organ systems, we need to know how our sympathetic nervous system is going to innervate. All right, so if you can get this done for tomorrow, this will make things a lot easier and smoother. You won't have to race to do your drawings and instead we can focus on talking about it and going through it that way. All right? Questions on that? All right, excellent. In that case then, because again, I know there's a little bit of time, but I don't want to start our first pathway and then have to take a break and come back tomorrow. So even though it's a little bit early, this is a good stopping point. So we will go ahead and stop here, and then we'll pick up with this picture, with this anatomy, and with this process tomorrow. All right, any other questions? All righty, then you guys have a good day, safe day, study hard, and tomorrow I will see you for our very last lecture. Hopefully you're as excited as I am.